IGCSE Biology 061, Paper 6, Variant 1, May June 2024, question number 3. And question number 3 is figure 3.1 is a photograph of a cross section of a persimmon fruit and which is magnified 2.2, and they draw a large diagram of the persimmon fruit shown in figure 3.1. We have to draw this persimmon fruit. So before drawing, we need to keep in mind some points, and those points are, first of all, we have to use the sharp HB pencil. HB pencil, and the other point is that the outline, the boundary line should be a continuous line. It should not be broken line. No double line. And inside you can see 1, 2, 3, 4, 5, 6, 6 seeds, large seeds you can say, and 2 seeds which are still closed. So all these points should be kept in mind when we will draw this figure. And the space you can see which is given to for this drawing, about 2/3 of this space should be used. As for example, when you are drawing in this way, it should be resemble with the picture in this way. When you are drawing, you have to draw properly. You can see our drawing will be a little bit changed as we have to draw it on screen. So there cannot be a proper drawing in this way, but you have to draw it properly, and you have to keep all these points which I have discussed. Now we are moving towards the next question, and the next question is line PQ is on figure 3.1 represents the diameter of the persimmon fruit. Measure the length of the line PQ on figure 3.1. Length of PQ is, I have measured this line, which is, let me show that line to you people. So this is the PQ line. I have measured this line. It is about 10.4 centimeter. So first of all, we will convert this line into millimeter. So because they have given this into millimeter, so we have to change this 10.4 into millimeter. So we will multiply with 10. When we multiply it with 10, then the answer will be 10.4, and if it is multiplied with 10, you can see the answer will be 104 millimeters. Simply, the other method is this. You can use this method as well, or simply you can multiply it with 10.4 with 10. So the answer will be 104 millimeter. Now, by using this value, we have to find out the actual diameter of the persimmon fruit using. Okay, so magnification for this formula, magnification has been given. See, the magnification is written over there. So directly we will use this formula, and it is 2.2. The magnification is 2.2, and length of the it is 104, and we have to find the actual diameter. So we will do in this way. We will divide 104 when we are going to find out the actual length. Then we will 104, we will divide it with 2.2. So the answer will be equal to 47.3. So this is the answer. We have to find the this actual diameter of the persimmon fruit. So in this way, we will calculate its actual diameter. Next question is, plants can make fat, starch, and protein. State the name of the reagents that can be used to test samples of plant tissues for starch and protein. You know, the starch can be detected by using the iodine solution. And you know, the iodine is yellow, um, yellowish brown, you can say, or yellow in color, or brown in color. Both of these colors can be used for this. Uh, and the brown color or yellow color is is changed into blue black and blue black, which shows the positive result, that is the presence of starch. If starch is present, then we will use the iodine solution. If iodine solution is changing from yellowish brown color to the blue black, it shows that starch is present. And for protein, we use the burette reagent, which is of blue color. So the if protein is present, the blue color will change into purple. So if blue color is changed into purple color, it shows the positive results, that is the protein is present. And the next question is, describe the method you would use to test a sample of plant tissue for fats. For fats, emulsion test is used. Emulsion test is used to find out presence of fats. For this purpose, we will add ethanol sample. If it is in the form of solution or in any other form, sample plus ethanol. We will add ethanol and then we will add water into it. And then we will shake thoroughly. Shake thoroughly, then cloudy appearance. Cloudy appearance or the formation of emulsion. Emulsion will confirm the presence of fats. Now we are moving towards the next question, and next question is, the enzymes pectinase is used to produce apple juice. Pectinase breaks down the cell walls in apple tissues. This increases the volume of juice that can be extracted from the apples. A student investigated the effect of the concentration of pectinase on the volume of juice extracted from chopped apples. The masses of 7 samples of chopped apples were measured. The samples were then put into the different beakers. 50 cubic centimeter of pectinase solution was added to 6 of the beakers. The concentration of the pectinase solution was different in each beaker. The beakers were kept in a thermostatically controlled water bath set at 37 degrees centigrade for 30 minutes. So it means the time was fixed and the temperature was fixed. Water bath temperature was 37 degrees centigrade and the fixed time was 30 minutes for all. The contents of each beaker were filtered through filter vapor and the liquid was collected in a miring cylinder. The volume of the liquid in each miring cylinder was recorded. State the dependent variable in the investigation described. And we have to find the effect of the concentration of pectinase on the volume of juice extracted from chopped apples. So it means the dependent value will be the volume of juice because we have to find out the effect of concentration of pectinase. So what will be the effect of pectinase on the volume of the juice which can be extracted from the chopped apples? So it means the independent value will be the concentration of pectinase, whereas we have to write the dependent variable. So the dependent variable in this case will be the volume of the juice. State 2 variables that were kept constant in this investigation. Already told you above when we were reading the question. So I had told you that there were two variables which were kept constant as the 37 degrees centigrade. So the variable which was kept constant, temperature, it was kept constant, that is 37 degrees centigrade, whereas the time was also constant, which was 30 minutes for all of these, and you can say the different concentrations. So in this way, we have this answers. Next, we are moving. The next question is identify a hazard in the method described in 3C and suggest a safety precaution to reduce the hazard. And students, here in this uh, question, uh, first hazard which we can write, that is the use of the pectinase enzyme. Use of pectinase enzyme, which is a chemical, and it can calm, it can harm the hands. So we can use gloves to save the hands. The student did not repeat the investigation and only collected one set of results. Explain why it is better to collect several sets of results. Yes, it is better to collect several sets of results because it will give more accurate results. So, to get more accuracy in results or to identify anomalous values. next question, question number 5, and you can see this question is this seventh beaker of the chopped apple did not contain the pectinase solution. State what should have been added to this beaker to make it a control experiment. So we have to keep the volume in all of these, you can say different beakers, 7 beakers. So in 6 beakers, we had added the pectinase enzymes, but in 7th, we have not added any pectinase enzymes. So here in this, we can add the water. Water can be used to add into the 7th beaker. So in this way, uh, we are moving towards the next question. And next question is, the results of the pectinase investigation are shown in table 3.1. And here you can see the 2 columns, percentage concentration of pectinase solution. So all these are the different percentage concentrations of the pectinase, which starts from 0.2, 0.4, 0.5, 6, 0.8, and it ends at 1. And the volume of the liquid collected per gram of chopped apple, and the liquid which was collected, this it starts 0.69, 0.77, and 0.81, 0.85, 0.92, 0.92. Last 2 values, which has the both concentration 0.8 and 1.0 has the same values. But as the percentage concentration of the pectinase solution is increasing, the volume is also increasing. The volume of the liquid which will be collected from the chopped apples, it is also increasing. So they are inversely proportional to each other. That is, percentage concentration of the pectinase solution and the volume of liquid collected from chopped apples is are directly proportional to each other. As percentage is increases, the volume is also increases. But last 2 values have the same volume collected from chopped apples. It means the highest concentration, which give the maximum amount of the liquid from the chopped apple, is 0.8, and 1.0 remains the same. Now we are moving towards the this question. Here you can see the next question. The sample of 150 grams of chopped apple was placed in a beaker with the 0.8% percentage solution. Using the information in table 3.1, calculate the volume of liquid collected in the measuring cylinder. Include the unit. So here we are going to find out the space for working they have given. So we have to this 0.8, the value at 0.8, the percentage. So what will we do? And we have to find sample of 150 gram. So we 0.92, we will multiply it with 150 grams. So the if we multiply it with them, then the answer will be 38, 138, 138. So it is the answer. Simply, we will use this 0.92, which is the highest, you can say, volume obtained at the percentage value 0.8. Now we have to take the 150 gram of chopped apples. So we will multiply it 150 because we have this value obtained after investigation. So when they have given us 150 gram of chopped apples, then we will multiply it with 150. So we are able to get the answer, which is 138 cubic centimeter. Now, the next question is plot a graph on line graph. We have to draw the line graph on the grid of the table in table 3.1. So students, here we are going to draw the graph. I will try my best to draw on screen. Uh, First of all, we have to take the values for this purpose. We need on one side, we have to take the percentage concentration of pectinate solution. So we will take this value on x-axis, percentage concentration, and on y-axis we have to take the, this is the independent value, and we have to take the dependent value because the volume of the liquid, it varies as the concentration is increasing. The volume is also increasing. So we will take this is the independent value, and dependent value, that is the volume of the liquid, volume of liquid of chopped. So we will take it on x-axis. So the the values start from 0 0.2. 0 0.2, 0 0.2. So in this way, 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So in this way, we have to count the 10. 0 0.2, 0 0.4, 0 0.6, 0 0.8, and 1.0. So on the other side, it's the value starts from 0.69. So we will take this value from 0.60 and then 0.70, 0.80. We will take it equal to 10 small squares. Students, you have to keep in mind that when you are going to draw all these, 0.92. So first value is on 0.2, and at 0.2 it is 0.69. So it is it will be it will be here. I'm drawing it on screen, so it is very difficult for me to draw the proper, but I am giving you the idea. At 0.4 and 0.5, 0.6 and 0.8 and 0.8. This is one. And 0.4, it is 0.77. So 0.77 can be here. And then 0.81, I'm only giving you, keep in mind, telling you again and again that I'm giving you the idea, because it can be drawn on paper. 0.9, and on 0.6 it is 0.85, which is will be here, and 0.8 it is 0.92, and on 1 it is also 0.92. they have the same value. Now we have to join these lines with the help of scale from this point to this point, from this point to this point, and this point, this point, this point, and this point. So just like this, we can get a graph on paper, good graph on paper. But you can see, little bit difficult for me to draw over on this page. Anyways, you have got the idea. And the last question is, describe the relationship between the volume of liquid collected from the chopped apple and the concentration of pectinate solution. Already told you that the concentration, as concentration is increasing, the volume obtained is also increasing. The volume of liquid, or you can say the juice obtained from from chopped is increased. But the last two values have the same volume of juice extracted or obtained. So it was all about the question number 3. Please like, share, and subscribe my channel.